Hello, everybody, and welcome to the first Friday, the end of the first week of the new year. We're going to talk about some RNA today, which stands for ribonucleic acid. But first, make sure that you answer up that bell ringer. You should have all of the week stuff done at this point now, so double check to make sure that is the case for everything. It's always good just to double check. So those will be going into the grade book later on today. Want to make sure you're starting off the first week strong. That are getting all your points for participation through there. We're going to look at the next part of our DNA RNA notes, which is number 3 on the notes. So without further ado, we can pull up those notes. There will be a copy of this in the Thursday folder. So they're going to be in there. So first off, we're going to talk about RNA. What is RNA? What's the deal with RNA? RNA stands for ribonucleic acid. You'll remember the other day we had DNA. That stands for deoxyribonucleic acid. So this is a same type of molecule for the nucleic acid part. It's just got the deoxy part removed from it. We're going to look at what that actually has to deal with here in just a little bit, because we're going to look at the differences between DNA and RNA, and then look at why we need RNA to begin with over the next couple of days. First off, DNA, one of the main differences between DNA and RNA is that DNA is double-stranded. I need to do this the other way so that the other side pops up, but we'll just run with it right now because we're doing it live. DNA is also have a deoxyribose carbon sugar, and then it also has thymine paired up with adenine. Those are the things that we covered on, I believe it was Thursday. No, it was Wednesday. Wednesday we covered it. My days are getting off because Monday, which is when I'm recording these, is not a part of our week because I'm just here. You're still at home. RNA, however, here's where the differences come into play. RNA is single-stranded. It's only got one strand to it. So it's just alone by itself doing its thing. It doesn't have its little dance partner with there. So it's riding solo with it. RNA. Single rider. It's got a ribose carbon sugar instead of the deoxyribose sugar, so it's got different sugar to it. Maybe it's a different molecule if it's a different sugar. We talked about a nucleotide has 3 bits to it. One of those parts is going to be different. And then it also has a new nitrogen base. It has what is called uracil. Usually we represent it as a U paired up to it and paired up to an adenine instead of thymine. So we have a new nitrogen base added to it as well. Thymines do not exist for RNA, just like this uracil didn't exist for DNA. It's only got one part to it there. So here's just some of those differences in picture form there. You can see the double strand on one side, single stranded on the other. I think the RNA looks more like the spiral staircase because it only has the one into it, whereas the other one's more of the twisted ladder. And I think staircases, I just think of like the one side, I guess, to it as it goes up. Some people disagree with me there for it. You can see that it's got thymine on one side, uracil on the other. And you can also see they've colored in the backgrounds a little bit differently on those little twisty parts. The DNA is orange there for the backbone, whereas the RNA has that reddish tint to it, maybe like a little brownish tint to it. Mainly to show that it's different. It's got a different part making up there. That's the different sugar going in. With RNA, the special thing about RNA that's different than DNA is that there's actually 3 different types of RNA. The first one we're going to look at, and these kind of go in order as far as when they show up. The first one we have is what is called the messenger RNA, which is usually just called the mRNA. And we lowercase the m, but the RNA part are all capitalized. So that is correct the way that I have written it there. Think of like the iPad. The I is small, but then you capitalize the P. That's kind of what we got going on here. The M is going to be small, whereas the rest of the RNA is capitalized. What this thing is that the messenger actually contains the DNA code that is going to deliver the message to a ribosome to eventually create a protein. The way how DNA is working and the way that it actually is the blueprint of everything is that DNA is going to create our proteins. And with that being said, the DNA code needs to get to the ribosome. The way that it gets to the ribosome is that it has to convert it into an RNA message first. So that is what our mRNA part does for us. tRNA stands for transfer RNA, and this is going to be the part that helps build the protein. The transfer is going to take after its name. It's going to transfer amino acids, which if you don't remember, because it's been a couple of months, amino acids make up proteins. It's going to bring in one amino acid at a time to help build up an actual protein for us. We're going to look at that steps over the next couple of days. So that's a little bit more involved of process. And then we got what we call RRNA, which stands for ribosomal RNA, which literally is what makes up the ribosome, which is why the mRNA has to be in the mRNA fashion, because the ribosome, which makes the protein, is literally made out of RNA, so it knows the RNA language to it. It doesn't understand DNA. It only understands RNA, which is why we got to get the message into the RNA first. And you can kind of see why we need it single-stranded here. I'll go back to this one here. Because as the message is being received, it knows how to pair up because of what we call the complementary message that is being sent there. So it's knowing what needs to connect up, what part needs to be brought in, what amino acid needs to build the protein because of that message and the order that that message is going to be in. So those are the three types of RNA, our MTRs, our message, transfer, and ribosomal there, all kind of mixing in. And that is going to be the thing that's going to help create a protein. That little ball structure there is going to be the protein. You got your that little pink little ribosome going on there. You got the messenger kind of feeding it the information there. You got your little tRNAs coming in through, dropping off the necessary parts in order to build it. So all of that to make sure that we have the proteins that are needed for us in order to do everything. And what those proteins end up doing, those proteins are then going to be used to build things inside of our body, from our different types of tissues getting together to our different organs getting together. I know we talked about cells going to tissues, going to organs. Some of those kind of connecting blocks and how it ends up knowing what a heart is based off of those proteins that are being made inside of the cell. Those proteins are being used to kind of help guide those actions to it. We got a little video from those Amoeba sisters again. I love them for this DNA, RNA stuff. They do a great job showing the visuals for it. So this one's looking at DNA versus RNA. I can't click past this, so yeah, that's the last thing. Ending off with the video there. And that's going to be our Friday notes for today. And as we have here, as it says here, there's some questions on Schoology, just like when we went through DNA. There's going to be a couple of questions on here. Here's our Schoology assignment. We open it up just like before. You got 3 attempts on it, 15 points possible. This one only has 8 questions to it. So you got your usual stuff that you can use your notes for. I've got this section here where I'm going to give you a statement, and you have to choose whether I'm talking about DNA, RNA, or both. So remember, they do share some similarities here. And basically what this part is, if you want to know what this has to deal with it, this would be like if we were to do a Venn diagram. This would be how we would do a Venn diagram. These would be the things that we would distinguish between. But because it's hard to do a digital Venn diagram, that's how we'll do it. A little multiple choice, you have to pick DNA, RNA, or both. That'd be like putting it on the DNA circle, the RNA circle, or right in the middle there. Then you got another question there. And a final question. Some of them have pictures to them, some of them don't. Yours may not be in the same exact way because I do have them shuffled about, so you may not have them in the exact order that you're seeing presented there. But you got 3 chances to get everything correct on there, so make sure that you're using that wisely to make sure that you're doing everything possible to get as many points there. Because that's going to finish our first week, and we got a good base knowledge of how DNA works, which is going to be important because as we travel into genetics, which is going to be next month, this is going to be the kind of crucial foundation that we're always going to be referencing. And even when we get into things like natural selection and things like evolution, this stuff will be things that we go back to because DNA is a driving factor for the rest of the year. Kind of like how we've always dealt back to cells in the first half of the year. This second half of the year, we're going to be delving back to DNA quite a bit. So we need to have a good, strong foundation in order to support ourselves as we learn more and more about what is happening with this little weird twisted ladder double helix shape going on with it there. But that's it for today. Make sure you get all your assignments done there. It's due on Monday. Hopefully on Monday I see you back, but if not, if you have any questions, do not hesitate to email me. If you want to set up a Google Meet, always available to meet, have those meets going during class if you would prefer to join us that way. Otherwise, I hope these videos are doing you well, and I hope you're staying safe and well there where you're at. And I will see you later. Adios. Bye.